Great pleasure to welcome you back from your tea break and to be able to introduce the last speaker today, who is Roger Kornberg from Stanford University. He's going to tell us about eukaryotic gene transcription, but Roger is the only one of the speakers that I've actually had personal contact with in the lab. So I might just throw in a few extra little observations here. The main one is when I came back from being a postdoc in America to start some independent work of my own in Cambridge. You know, we were working hard and so on. And in our division, I was in one lab and Roger was in the next lab. And we would work, you know, all the day and so on and so on. And then the hours would go by and gradually everybody would disappear until only Roger and I were left. And then, you know, I would say, oh, I think it's 2:15 a.m. I said, I think it's possibly time for me to go home. And Roger would say, oh, so early. So I'm sure that now he has a group of other people who help him. I'm sure he still works beyond 3 a.m. in the morning. Anyway, Roger.
So thank you, Richard. Thank you for the invitation. I'm glad to be here. And I would like to tell you about two different lines of work, both of which relate to chromosome structure and transcription. So I'll speak first about work done by Yali Lorch and Shigeki Nagai, also Barbara Mayer Davis, not listed here. This is work that concerns nucleosomes, the basic unit of chromatin, and transcription. And most of what I present on these lines has been published. Then in the second part of the hour, I'll turn to work on chromosome structure, that is structure beyond the nucleosome. That is the work of Andrew Beal, Pierre-Jean Mattei, and Maya Azubel. And none of this work has yet been published, at least, I should say, despite our best efforts, but we'll keep trying. Now, when I put forward the idea of the nucleosome some 45 years ago, and it was the result of that work to which Richard refers, so it was an idea that came about from work that I did at the LMB in Cambridge at the same time as Richard and I were both there. When I put this idea forward, it was apparent that wrapping DNA around a set of histone molecules in chromatin or in the nucleosome would interfere with many DNA transactions. And only a decade later, Yali Lorch and I showed that wrapping promoter DNA in a nucleosome would prevent the initiation of transcription in vitro. Only a year later, Michael Grunstein and his colleagues showed a similar inhibitory effect of the nucleosome upon transcription in yeast cells in vivo. So the nucleosome then, as indicated here, serves as a general gene repressor. It prevents expression of all the many thousands of genes in eukaryotes except those whose transcription is brought about by specific positive regulatory mechanisms. And so for this reason, I regard repression by the nucleosome as the first principle of eukaryotic gene regulation. Now, it's not surprising that the nucleosome is generally viewed as a packaging device to serve principally that role. That is not surprising it's viewed as a component of chromosome structure because the histone proteins represent about half of all chromosomal protein. The numbers, however, argue against a primary structural role of the nucleosome. Wrapping DNA in the nucleosome achieves a reduction in length, a contraction of the DNA by only about a factor of 5. But the contraction of length in the most condensed state in mitosis is a factor of 10,000. And I think it's scarcely believable that half the mass of the chromosomal protein is used to solve less than 1% of the DNA packaging problem. And in fact, as I'll discuss later when I turn to the subject of chromosome structure, it is condensins that are conserved from bacteria to humans that are believed to be responsible for the contraction of length of DNA in chromosomes. Now, a first indication of the primary role of the nucleosome actually came from work done about 20 years ago by Kaplan and Winston. They showed that a mutation in the SPT6 gene of yeast, the SPT6 gene is believed to be a histone chaperone, a mutation in that gene would cause a depletion of nucleosomes and also aberrant transcription. What they found was transcription initiation within open reading frames of genes. More recently, Fred Winston and his colleagues have extended that work. And what they have shown is not only are there more transcripts initiated within genes, that is to say intragenic transcripts in the SPT6 mutant, and more even than genic transcripts, only 4,000. But further, that so much of the transcription machinery is diverted to intragenic transcription that the normal genic transcripts are actually downregulated, expressed to a lower extent. So what is apparent, I think, from what I have just told you, nature invented the nucleosome to contend with the promiscuity of the transcription machinery. The nucleosome is not only a general gene repressor, it's a general transcription repressor. Now, the logic for the design of the nucleosome for this purpose, I think, is really quite straightforward. The only way of preventing promiscuous or widespread transcription is clearly by covering up the relevant DNA. Now, the proteins that bind DNA that we're most familiar with, activator and oppressor proteins, are typically on the order of 50 to 100 kilodaltons, and they cover up about a dozen base pairs of DNA. But the alternative, instead of having the protein cover the DNA, having the DNA cover the protein in the nucleosome, achieves an order of magnitude greater efficiency. Now, in the nucleosome, 146 base pairs of DNA cover 100 kilodaltons of protein. Now, we know from our structural studies of the transcription machinery that the nucleosome prevents assembly of a transcription complex by simple steric exclusion. The nucleosome and the transcription machinery cannot simultaneously occupy the same DNA. Nevertheless, as I will now explain on the basis of work that, as I say, has recently been published, a nucleosome covering a transcription start site can actually enhance transcription initiation. So I'll begin by reminding you of the composition of the transcription machinery. Some 30 years ago, my colleagues at Stanford, also Rader and his colleagues at Rockefeller, also Ronald and Joan Conaway, then at the Oklahoma Medical Research Foundation, identified the components of the polymerase II transcription machinery. The polymerase, of course, and as many of you know, is capable of unwinding the DNA double helix and synthesis of RNA, but alone incapable of recognizing a start site for transcription or initiating the process. And it is for those essential steps that a set of so-called general transcription factors that go by the letter names B, D, E, F, and H are required. Now, when we identified the machinery as summarized here, it was widely believed to be not only necessary but also sufficient for transcription in all its forms. That is to say, not only able to support the initiation of transcription, but also the regulation of the process. Nevertheless, in 1990, my colleagues identified an activity in a yeast extract that we referred to as mediator, which we found was required for a response of the system composed, as I showed you in the previous slide, to the action of regulatory molecules such as activators and repressors. It was a few years later when my colleagues Kim and Bjorklund succeeded in isolating mediator in homogeneous form as a 21-protein million molecular weight complex. By now, it's generally accepted that mediator is universal. It receives and it processes regulatory information for all RNA polymerase II transcription in the entire range of eukaryotes, from yeast to human cells. So nearly 60 proteins then assemble in a complex the size of a ribosomal subunit, so 3 megadaltons, at every RNA polymerase II promoter prior to every round of transcription. Now, all the work done to identify this machinery was performed with naked DNA as a template for transcription. We wanted to extend the work and perform such experiments with cellular chromatin, with genes as they occur in vivo. Many studies had been performed studying transcription of DNA associated with purified histones, but it seemed important to extend the work to actual cellular chromatin. In many cases, we don't know the precise post-translational modification state of the histones at promoters in vivo. Neither do we know what may be the additional non-histone proteins present at one promoter or another. And so for these and other reasons, we pursued the isolation of a gene as chromatin in the manner that is shown in this slide. So it begins with placing recombination sites on either side of a gene, in this case, the Fo5 gene of yeast, followed by expression in the yeast cells in vivo of the recombinase which acts at those locations to excise the gene as a chromatin circle released in this manner from all the rest of the genomic material. And then by an affinity method, we were able to purify the gene circles. We could enrich them about 200,000-fold to near homogeneity. Now, the transcription of these gene circles studied by Shigeki Nagai was in keeping with what I have told you. So transcription with polymerase and the general factors was undetectable from genes in chromatin, whereas naked DNA extracted from the purified chromatin gave a measurable level of transcription. What Shigeki found was that addition of 5 additional proteins enabled now initiation and transcript elongation of the Fo5 transcript from the gene circle. Now, we had thought that if we could somehow reproduce the gene activation process with a chromatin circle, we might ultimately achieve a level of transcription comparable to that obtained from a naked DNA molecule. But instead, as you can see here, the level of transcription from a chromatin template was much higher than that from naked DNA. I just realized I didn't turn on the microphone. Could I be heard in the back? I'll turn it on anyway. Now it'll be too loud. What we found was that the ratio of transcription from chromatin to that from naked DNA was even greater as the concentration of all of the transcription proteins was reduced, reaching a level at the lowest concentration that we measured of nearly 20 times greater than that from a naked DNA molecule. So apparently, chromatin is more effective at capturing the transcription proteins than naked DNA. Or in other words, chromatin must in some way interact with the transcription proteins. We imagine that it's possibly a nucleosome, but then the question becomes, which nucleosome? The answer could be found from the experiment that begins in the following slide. Sorry, let me back up, because I forgot to tell you that transcription in this complete system also requires acetyl-CoA as a substrate for the histone acetyltransferase of the saga component of the machinery. Of course, acetyl-CoA is not required transcription from a naked DNA template. So just to summarize what I've told you then, the level of transcription, as I've said, at least 20-fold greater than that from chromatin than that from naked DNA. I've indicated that that implies an interaction between the transcription proteins and chromatin, presumably nucleosomes, and finally that it's dependent on histone acetylation, and I haven't shown you, but also histone methylation. So this then is a summary of studies done many years ago by Wolfram Hertz and his colleagues analyzing the state of the Fo5 promoter in GNET chromatin in yeast cells, so in vivo. And what he showed is that in the repressed state, the gene is not surprising, and the promoter not surprisingly packaged in an array of nucleosomes which cover the transcription start site, the promoter including the TATA element for the initiation of transcription, and also one of two regulatory sequences bound by the Fo4 gene activator protein. In the activated state, following induction of Fo5 transcription, two nucleosomes are removed, and the one that covered the promoter and start site slides to expose the promoter but still covers the start site. And we could exploit this observation to discover which nucleosome is the target of the transcription proteins to enable enhanced transcription by placing, again, sites for the recombinase, one upstream of the gene, but the other not at the downstream end, but rather immediately adjacent downstream from the promoter. So a gene circle isolated in the repressed state would bear only 3 nucleosomes, and one isolated from the gene in the active state would contain a promoter and only a single nucleosome covering the transcription start site. And what Shigeki Nagai found was still enhanced transcription from the activated gene circle bearing a single nucleosome covering the transcription start site. So on that basis, we believe that a nucleosome covering the start site of transcription serves actually as an important component of the transcription machinery. Now, with that, I will turn to the generality of this result. It isn't limited to Fo5. This again summarizes what I told you about the results from Wolfram Hertz. This is a picture which turns out to apply to every transcribed gene in yeast. So here you see a type of analysis that is done by many labs. It happens to be a result computed by a student of mine. It shows the results of micrococcal nuclease digestion to cleave between nucleosomes, and then high-throughput sequencing to discover where the cleavages occur. And so what is plotted on the vertical axis is the likelihood of finding a nucleosome, and then what you see is the distribution across about 100 kilobase region of the yeast genome. And what it shows is a more or less monotonous array of nucleosomes punctuated by occasional absences. And these occur immediately adjacent to start sites of transcription. If we zoom in to look at an example in the next slide, what you see is indeed a nucleosome covering a transcription start site, a so-called +1 nucleosome, and adjacent one of those exposed regions, a so-called nucleosome-free region, or NFR. The promoter and also sites for binding regulatory proteins are located exposed within the nucleosome-free region. Now, with that, I'll turn from chromatin and transcription. I'll turn from the nucleosome as an element of chromosome structure to structure beyond the nucleosome, so-called higher-order structure of chromatin. This is a challenging problem that has been studied and not a whole lot of progress made for many years. And the reason is that chromosomes are the largest, the most complex, and probably the most dynamic of cellular organelles. Also, the problem is that chromosome structure is in some way paradoxical, because mitotic chromosomes, which I'm sure everyone has seen or is aware of from study or pictures, exhibit an approximate uniformity of shape. They're all roughly cylindrical. And in any picture that you see, they all have approximately the same diameter. And yet they have a highly variable composition of the underlying chromatin fiber. So macroscopic order is somehow imposed on microscopic disorder. Now, I mentioned before that condensins are important for the 10,000-fold contraction and length of DNA and mitotic chromosomes. Condensins are, especially from recent work, believed to form loops of chromosomal DNA. Now, the idea of a loop is not new. It was known for over 100 years from classical cytogenetics, from observations, for example, of lamp brush chromosomes, in which loops of chromatin radiate from a central axis. The problem is that loop formation achieves linear contraction, but no condensation. Condensins contract DNA, they don't actually condense chromosomal material. And clear evidence of this distinction comes from a study that was done by Bill Earnshaw, who showed that the destruction of condensins in mitotic cells does not prevent condensation. The chromosomes still condense, but they condense in a single formless mass. No mitotic chromosomes can be observed. So contraction of length and chromosome condensation are clearly distinct processes. Now, the challenge for structural studies is that existing methods, at least physical methods, are not readily applicable. So X-ray diffraction of mitotic chromosomes has been uninformative, and X-ray diffraction has in general failed to reveal much information about so-called higher order structure of chromosomes because the chromosomal material lacks long-range structural order. Electron microscopy has been unrevealing because chromosomes are so dense. The density of mitotic chromosomes is too great for transmission of an electron microscope beam. So, for want of a better approach, we decided to investigate the forces responsible for chromosome condensation. And in particular, we developed procedures for quantitation of the condensation behavior. So for this purpose, we adsorbed individual mitotic chromosomes to specially coated glass slides and then perfused with solutions of various composition. The chromosomes were, for the most part, observed with DIC, with differential interference contrast optics. And what you see here is an example of a human helichromosome, which, after perfusion with a dilute solution containing only 5 millimolar tris buffer and 2 millimolar potassium chloride, swells to about 10 times its original size. Now, it's long been known that chromosomal material, and chromosomes in particular, will swell and contract on raising and lower ionic strength. But the observations made in the past have been mostly made on just clusters of mitotic chromosomes or isolated chromatin. And the observations have been just that, simply observations, qualitative, nothing more. We wished to make precise measurements on individual chromosomes and obtain quantitative information. For example, as shown here, we began by just measuring the length to width ratio of the many different helichromosomes, comparing the length to width ratio in the absence of a polyamine, a naturally occurring polyamine actually associated with chromosomes called spermidine, or in its presence at a substantial concentration. And what you can see is a plot with a slope of 1. The length to width ratios remain unchanged, which tells us that the swelling that you see here is isotropic. It occurs equally in all directions. In this particular case, the chromosome was imaged with the use of a fluorescent dye, but we learned to avoid that and instead used DIC optics, as I've mentioned, because it turned out that the dyes themselves can cause condensation. Now, it turned out to be particularly advantageous to switch to differential interference contrast optics because it reveals the chromosomal material directly, and in particular, in a chromosome following swelling, it revealed a central filament, not visible before the chromosome was expanded in this way. That central filament must represent the scaffold that was described by Ole Lemley in the past, which he reported from electron micrographs, which some of you may have seen. It appeared there as an irregular mass after removal of all the chromosomal proteins. I thought, and many others were concerned whether the scaffold really existed or whether it was just a remnant of extraction. But with this experiment of our own, certainly I'm convinced, seeing it now in the native state, that the scaffold is a meaningful structural element. And it's mostly proteinaceous because DNA suggestion to remove all detectable DNA has no effect upon its structure. So as I've said, chromosomes could be decondensed in 5 millimolar tris 2 millimolar potassium chloride, and they could be recondensed by the addition of spermidine. And that process of decondensation and recondensation could be repeated over and over again with no apparent loss of morphology. Now, in these experiments, the perfusion with the dilute salt solution had to be, as I'll tell you later, performed very slowly over a long period of time in order to record the behavior of individual chromosomes over such a long period of time. We had to deal with a variety of problems: instrument drift, inhomogeneous illumination, fluctuations in lamp output, and other sources of movement, variation of intensity. And not only that, but the outlines of the chromosome in the decondensed state, from which we wish to measure its area, were barely visible. And this posed a challenge for automated data collection and processing, which is essential if we wanted to average the behavior of hundreds of individual chromosomes. Andrew Beale, who I mentioned, wrote many thousands of lines of computer code to correct for spatial and intensity variation, and also to solve the problem of image recognition in the fully decondensed state. And his automated procedures then allowed the collection of a very large number of images, hundreds of thousands of images, correction for spatial and intensity variation, selection of particles, and finally solution of the problem for area determination in even the most decondensed state. And so with the use of those methods, Andrew and P.J. Matai have, in the first of the series of experiments, and I'll show you, plotted the degree of condensation as a function of the concentration of the polyamine added for repeated cycles, 1, 2, 3, 4, and so forth, of decondensation and recondensation. And you see that the recondensation behavior is exactly the same every time. And then they measured the absolute degree of condensation in the fully condensed or in the fully decondensed states. And you can see that it's the same for every cycle. So we conclude that to the resolution of this analysis, the chromosome condensation that we produce and observe is fully reversible. I think the recovery of the starting morphology following decondensation to 10 times the starting size and recondensation is really remarkable, because the chromosome doesn't simply collapse, recondense to a formless mass, but rather it regains its original size and shape. And I think the memory of the morphology must reside in that central filament, and that the morphology of the mitotic chromosome at this level of analysis is simply explained by condensation against a central filament, a central axis. Now, the results of further experiments, looking in more detail that I'll show you in a moment, may seem to you unnecessarily detailed, but they really come to the crux of the analysis, and so please bear with me while I explain. I think the important point will become apparent at the end. So what you see here are a series of condensation profiles for a set of polyamines that have a 1, 2, 3, or 4 positive charges at neutral pH. And the data points, each data point represents an average of anywhere from 208 to 504 individual chromosomes. Now, the lines through the points represent least squares fit to the Hill model from which an apparent dissociation constant of the individual amine compound can be derived. And then the logarithms of those dissociation constants correspond, of course, to binding energies. And what you see is that there is a linear dependence of binding energy upon polyamine charge, except for the monoamine, which is not surprising because, as you probably know, monovalent salts don't associate with one another but remain fully dissociated in aqueous solution. And now I'll explain why what may seem like a detailed exercise is so important. The theory for condensation of an ionic hydrogel was developed by physical chemists 70 years ago, beginning with Paul Flory, important contributions made by Aharon Kachalsky-Katsir and others. And we could adapt that theory for application to chromatin and to the chromosomal material. And the basic idea of the theory is simple. It is that swelling is driven by osmotic pressure, and that the pressure draws water into the hydrogel until it drops to 0 at equilibrium. The osmotic pressure is due to the tendency of the material to dissolve and swell into the solution. It's due to what is called the Donan effect, which is the tendency of ions to enter the gel, bring water with them to neutralize the charge on the gel. And those two effects are opposed by the natural elasticity of the material. Now, what was derived over the years were closed-form expressions for each of these terms. For the dissolution or mixing term, the elasticity, and also the ionic term, which goes well beyond what I've shown you here. The point of it all is that these are closed-form expressions, and we could calculate after adapting to chromatin. There are many ways to show you the results, but the simplest is what is in this next slide. It is a remarkable agreement between theory and experiment, for example, for computing the binding energies of the series of amines to the hydrogel and the dependence on polyamine charge. I could show you other evidence of the agreement between theory and experiment. I think the most striking is something which we didn't expect. So with a theory in place, we could explore conditions that were not readily or so easily done experimentally. And the next slide shows you something that the theory led us to expect that we thought could hardly be true. So the blue curve shows the results from theory, and in particular it shows that decondensation, as one lowers the ionic strength to millimolar, occurs in the manner that we anticipated. But then the theory predicts that if you lower the ionic strength or the concentration of salt further to micromolar, that the chromosome will contract again and regain its original size. Now that we thought was bizarre and unlikely to be true, but it was borne out by experiment, as shown by the data points that are listed here. So that, I think, gave us the greatest confidence in the application of the theory, and then from that we could conclude that the chromosomal material is indeed an ionic hydrogel. Now, the classical theory, amine field theory, as I say, developed by, originally by Paul Flory and expanded by others, goes beyond to a further prediction that has particular physiological significance. The theory predicts that ionic hydrogel will exhibit critical behavior It predicts that under certain conditions, the gel will collapse. This is a change of state that the physical chemists refer to as a volume phase transition. The classical manifestation of a thermodynamic first-order phase transition, as some of you may know, is hysteresis. And that we indeed observed is shown in this slide. So a different path for decondensation than for recondensation, here in response to varying the pH, or I could have shown you spermidine or what have you. So hysteresis, as I show you here, is striking evidence, really a proof of a volume phase transition of the chromosomal material. And from this, we conclude that the structure of a mitotic chromosome derives from two simple organizing principles. The first, a linear configuration, as I have mentioned already, of loops of the chromosomal material. And second, of volume phase transition of a chromatin hydrogel. The cylindrical shape of the chromosomes and their uniformity of diameter then are simply due to minimization of surface area, driven, of course, as I've said, by electrostatics and by elasticity along a linear axis. Now, condensation by collapse, that is to say by a volume phase transition, resolves what I told you was the central paradox of the mitotic chromosome, namely the organization of a heterogeneous fiber in a regular structure. Collapse, of course, is compatible with local variation in the overall condensed structure. A further point that I won't go into detail, but introduces the next point of my presentation, is that the breadth of a decondensation-recondensation hysteresis loop is a measure of structural disorder. The breadth of the hysteresis loops that we observe is very substantial. The mitotic chromosomal material evidently adopts heterogeneous and disordered conformations in both condensed and decondensed states. And now I will conclude by showing you direct evidence for such disorder. So I already mentioned the problem of electron microscopy of chromosomes due to the high density of the material. We could, and in particular Maya Azubel, could exploit the decrease in density on decondensation, reversible decondensation that I've mentioned, to obtain images of the chromosomal material by cryo-electron microscopy, in particular by cryo-EM tomography. And what I show you in this next slide is a slice through a cryo-EM tomogram of slightly, only twofold decondensed mitotic chromosomal material. And what is really striking in this slice through the tomogram, already you can see many examples of nucleosomes, apparent often as double-layer discs, and in some instances of the linker DNA that connects between them. Now, as I've said, this is a two-dimensional view of a three-dimensional data set of a tomogram, and one could, in studying, as Maya did, the three-dimensional data set, find a connection, linker DNA connecting every single nucleosome. And in that way, she could trace the connectivity from one nucleosome to another and thus determine the trajectories of chromatin fibers. And three examples are shown here where, as I say, by going back and forth through the three-dimensional data, she could obtain and define unequivocally what was the connectivity. The first thing that is noteworthy, there is no regular organization. In fact, none of the structures of chromatin fibers that we've discovered in this way resembles structures that have been proposed on the basis of crystallography or cryo-EM of reconstituted material or previous studies of mitotic chromatin. This particular tomogram contains 520 nucleosomes. They're all shown here in a view from which I've omitted the linker DNA just for ease of viewing, but each of the different colors represents nucleosomes in a separate chromatin fiber. The disorder to which I alluded, coming from or anticipated from the width, the breadth of the hysteresis loops is plainly apparent. So here there were 520 nucleosomes identified in the tomogram, every one connected by a linker to another except for those at the boundary where, of course, there's no conductivity, no linker involved. It's also noteworthy that the average length of the linker could be calculated. It corresponds almost exactly to the length of linker DNA that was measured biochemically by nucleus digestion of mitotic helicell chromatin. So in this case, there are 18 fibers. They contain, in this tomogram, anywhere from 10 to 50 nucleosomes. And as I've mentioned, the chromosomal material in this region was only decondensed by about a factor of 2, so very little different from the native material. We can go on now to identify by the use of DNA sequence-specific binding proteins tagged with dyes or with heavy metal complexes or what have you, specific DNA sequences within such a tomogram. And then, because we can trace the fibers from one tomogram to the next, follow the path of the DNA and understand the structure of the chromosomal material through a substantial region of the chromosome. And in this way, to investigate the structures of genic and also intragenic chromosomal material. And I hope to return one day and tell you about the results of that analysis. Thank you for your attention.
So it's the beginning of a new view of chromatin, but I'm sure the audience will have questions. I'll ask one just as a warm, Venki, there's one, yeah.
So why does it seem to need a nucleosome covering the start site to activate transcription? That seems counterintuitive.
Exactly. So I think actually it serves as a repressor even in that location so that no adventitious transcription will occur unless a regulatory protein is bound in the nucleosome-free region. In a way, it's what you could call two-step authentication. So you have to unfold the promoter, expose the NFR, but still you protect the start site and prevent the onset of transcription except in the presence of a regulatory molecule which must now bind to its cognate site in the NFR, recruit mediator, and the rest of the machinery.
And then remove the nucleosome.
And then what I believe we have some evidence to indicate that the nucleosome will then be removed through the translocase action of TF2H, the one member of that set of transcription proteins which is a DNA-dependent ATPase helicase, which, as you know, unwinds the DNA to allow the initiation of transcription. But it is, like all helicases, actually a translocase. And the evidence that we have to this point would indicate that it is responsible then for the disruption of the +1 nucleosome.
Spermine is no.
Wait. Yeah.
So,
You have to wait, yeah.
wait. Thank you. So spermine can condense DNA. So in your spermidine or
any ion. And but it goes beyond that because it isn't only that the naturally occurring cations are abundant in all organisms and contribute to DNA condensation, but also any protein, even at a remarkably low concentration, will drive condensation. In fact, what we find is that the condensed state, that of the most condensed form of the material in the mitotic chromosome, is the natural state of chromatin. It is the default state. We used to think it was a question, how do you drive condensation? I told you at the beginning, we were looking for the forces that cause condensation. Turns out the problem is the opposite. Condensation always occurs. And the unanswered question is, how do you drive decondensation? Now, in retrospect, that makes perfect sense because most of the chromosomal material is condensed most of the time. 80 to 90% in interphase remains condensed because those genes or relevant DNA sequences are not transcribed. So in retrospect, it makes perfect sense that it would be decondensation, which must be selectively brought about by mechanisms that remain to be determined.
So at the bottom of the curve, when it's mostly condensed, this is the condition that...
That is the condition in cells at all times, in the absence of a mechanism that we don't yet know that drives decondensation.
I see. Thank you.
2 rows back, yeah.
Do you think that in the cytoplasm you have such changes in ionic strength and pH?
No. I don't think that it's driven, I don't think that decondensation, which I mentioned is the question, is driven by changes in pH, ionic strength, or what have you. It's more likely a consequence of modification of the chromosomal material itself to alter the charge, or patterns of cross-linking, or what have you. Indeed, we know that there's extensive charge modification due to acetylation of histones, methylation, phosphorylation, what have you. But it remains to be shown directly what modification or process of that kind is actually the one that drives the decondensation of the relevant regions in the interphase nucleus.
Couple of questions. One is that the central scaffold you showed, what do you think is composite there?
That's a great question. So what Hirano and Mitchison, and in later work, I think Earnshaw and others, Lemley, showed years ago, there are 2 principal components that remain after extraction to produce what they called a scaffold, which I didn't used to believe until I saw it here. They are condensin and topoisomerase 2. There are other proteins present at a lower level. Those are clearly the predominant components. I think finding the structure of that scaffold would be a really great thing to do, and we're trying. We have some preliminary diffraction information, but nothing really to share with you at this time.
Second question is that you showed that to get the 20-fold increase within chromatin, you need quite a few chromosome remodelers. What do you know those rows are for?
So the additional proteins that are required, which I didn't take time to mention because I was in a hurry to get to the end, those additional proteins that you need beyond polymerase and the 5 general factors are an activator protein, which is, in the case of the FO5 gene, FO4, the mediator, which I've already mentioned, the SAGA complex, which is a 2 million molecular weight complex, which contains, as I mentioned, a histoneacetyltransferase, contains a subunit that recognizes a methylated lysine residue in H3 at position 4, and many other components whose roles I don't know, some of which are shared with the TAF complex and TF2D. And also a remodeler complex, which could be either the switch-SNF complex or the RISC complex in these experiments.
But you don't, do you have a hypothesis of what it might be doing in terms of activation?
What roles do they all play? Look, in the case of SAGA, we think that, there's, as you may know, the +1 nucleosome has been shown to be extensively modified, post-translationally modified. The histones are highly acetylated. They also bear that H3K4 methylation mark. SAGA recognizes the H3K4 mark, and it may play a role in the acetylation. Mediator, we think, is important because it actually interacts directly with RNA polymerase and actually brings it to the promoter. The remodeler plays an interesting role. It is not there to remove the nucleosome, which is what you might imagine. I mean, the RISC complex, which we've studied for many years, we mostly know for its capacity to remove histones from DNA. But Steve Henikoff showed that actually there is a stable complex of RISC with the +1 nucleosome. And Yali Larson and I showed that the stability is actually due to the acetylation, which in the presence of which risk fails to remove a nucleosome. But the point about the a stable association with risk appears to be a partial unfolding of the +1 nucleosome. In the complex with risk, it appears that the DNA is partially unwrapped, and that may facilitate the answer to the question that Venki asked about the mechanism for removal, which occurs on invasion of the +1 nucleosome by the TF2H translocase.
So I have a question as well, Roger. So my question is, in the choice of twofold decondensation to show us, presumably you could have chosen 1.5, sixfold, eightfold, and then would the length of the linker have changed? And did you pick it so that it would agree nicely with the, or no, why did you pick twofold?
Okay, so we wanted to decondense minimally so as not to disrupt what structure may be present by stretching the material. And in fact, in a chromosome that is decondensed manyfold, the decondensation occurs in a heterogeneous manner. Some parts remain condensed, some fully decondensed. But the remarkable thing is that the length of linker in every region has the same average value as what I told you. So that isn't, I worry what you just asked. It is not a consequence of stretching. It turns out that the linker DNA, and this is, well, there's much more that we could discuss about that, but the linker DNA appears to be that precise, or that average length, which is interesting because the core particle of the nucleosome was identified by nuclease digestion, solved by crystallography, but we never knew if it was actually precisely the relevant form in chromosomes in cells. And it emerges that the core particle is the relevant structure from that analysis.
If there are no other questions, let's say thank you again to Robert.